Hi, my name is Aishi, and welcome to part 2 of our lecture on apoptosis. Now, previously we were talking about apoptosis in the embryonic stages of C. elegans, which is a nematode, and it's this awesome model organism because it has very few cells, and we know exactly where each of the cells comes from. Now, what geneticists have discovered are these two genes, two key apoptotic genes, SED3 and SED4. SED stands for cell death. And these two apoptotic genes encode proteins that are essential for apoptosis. What they also discovered was that these proteins, SED3 and SED4, are continuously present inside the cell. However, they're just inactive. It's not like all cells are constantly undergoing apoptosis. That's not possible because they would be dead. But they're constantly present and inactive. They're usually in their inactive form. And what this shows us is that the regulation of these of apoptosis occurs at the protein level rather than the gene level. Now, SED3 and SED4 are normally kept inactive by this protein in the outer mitochondrial membrane called SED9. And SED9 is this master regulator of apoptosis. When SED9 is active, it stops SED4 and SED3 from becoming active. So it keeps these two proteins inactive. However, when a death signaling molecule comes and binds to the receptor, this causes SED9 to be inactivated. And when SED9 is inactivated, SED9 is unable to inactivate SED4. So now SED4 is active. Now SED4 goes and activates SED3, and SED3 is a protease. It goes and it cuts a lot of things, and it activates other proteases as well as nucleases. And this leads to a bunch of changes in apoptotic cells, and this leads to cell death. Now, what we just covered is only one pathway of apoptosis in C. elegans. In humans and other mammals, there are several different pathways, all of which can lead to apoptosis. And the pathway that leads to cell death really depends on the type of cell and also the signals that are available that will trigger apoptosis. One major pathway that is involved has a lot of mitochondrial proteins that come together and form these pores in the mitochondrial outer membrane. And what this does is it causes proteins to leak out from the mitochondria, as well as other components of the intermembrane space to leak out. And in fact, cytochrome C, which is normally involved in the electron transport chain in the mitochondria, if you remember, cytochrome C will actually come out of the mitochondria. And when it's found inside the cytoplasm, it will act as the cell death factor and trigger apoptosis. Now remember SED3, SED4, and SED9 in this nematode C. elegans. Well, mammals also have the same source of proteins, right? These are essentially just relay proteins that are capable of transducing the signal. And what these relay proteins do is essentially what we covered before. They help pass the signal along, and they also integrate signals from a variety of sources. It's sort of like crosstalk between pathways all over again. It can get information from a variety of receptors that might trigger apoptosis. And often the signal for cell death will come from outside the cell. Remember the example that we just covered where you had that little red ligand that binds to the blue receptor and that causes cell death? Well, the signal for cell death can also come from inside the cell. For example, if you have too much DNA damage and you can't repair it, or if you have too much protein misfolding, then the cell will tell itself to kill itself. And like we've covered before, a lot of stressors can cause cells to die. And this is often, and cells will often sense that, it's under stress, like things are wrong, proteins are being misfolded, blah, 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 and it will choose to kill itself.